Today we are going to discuss about our fifth topic of this week. So we have discussed about blue white screening, we have discussed about insertional inactivation, then red white inactivation, then star activity. So now we are going to discuss about E.COLI, LAC and T7 promoter based factor. So this is our today's goal. So although I have prepared a detailed notes regarding the E.COLI LAC T7 promoter base vector, but it will be very easy to understand if we are going to use some diagrams. So here remember the one thing, that actual goal here is to produce protein in large amount, which is usually is of low abundance. Okay, so here we are going to produce this. So as I have told you that we are going to produce large amount of low abundance protein. So here our actual goal is this. So for fulfilling this goal, we are going to use E.COLI, LAC and T7 promoter based vector. Okay, so what can we do? So first of all, here we are taking the example of protein GCSF. Okay, that means granulocyte colony stimulating factor. This is the protein that we are going to synthesize. So first of all, we can easily determine the mRNA that is responsible for the translation of GCSF easily. Okay, so now it is easy to produce the cDNA from that mRNA. It is easy. We can do this with the help of reverse transcriptase enzyme. So now we are going to use this cDNA to produce protein GCSF. That is our actual goal. So and another important thing that we need to have for performing our goal is a plasmid with appropriate genes, I should say. That means the plasmid should be genetically engineered plasmid who have the ability to express the protein encoded by cDNA large amount in the host. Okay, so that means we want a well genetically engineered plasmid so that we can have our protein in large amount along with the cDNA that ultimately lead to the production of the protein. Okay, so that is the idea. So what we need to do, first of all, we have isolated cDNA. I have already discussed with you that how we can produce cDNA from mRNA during the cDNA library lecture. If you do not know how we can synthesize from mRNA cDNA, you can easily go back to this video and watch. So and here the characteristics of the plasmid that we are going to use should be contain a primary selection marker so that we can distinguish between the transform cell and non-transform cell. Along with there should be a origin of replication. Okay, then the multiple cloning site should be there, so that here we are going to insert our gene of interest. And along with in the multiple cloning site, you can conclude if you want to use blue white screening for your secondary screening procedure, then you can simply insert a omega peptide or alpha peptide. Because remember one thing that I have forgot to mention in case of blue white screening is that the gene of interest that we are going to use should not be too much disturbed. Okay, that is why in case of alpha complementation that we are using, here we add alpha with gene of interest, right? So this is technically and ethically not a right thing, because it will damage your gene of interest simply. So that is the reason why instead of alpha complementation we will go for insertional inactivation. So that means we are going to put a omega peptide, or you can say alpha peptide. We have discussed about that LAC gene consists of two peptides, one is alpha and another one is omega. So we will prefer omega, so omega peptide on the MCS. Okay, so that is the idea. So now we have prepared our plasmid vector well. Now we will simply treat them with restriction endonuclease so that we will generate a opened in plasmid for our cDNA insertion. Now before the insertion we have to modify our cDNA. Why we are modifying this cDNA, I am going to discuss with you within a minute. So the modification is, we will going to add a small or short stretch of nucleotide. Okay, you just have to remember this. So why we have to add this, and how much amount of nucleotide we need to add. So remember one thing, we will add the short nucleotide in such a way so that after the translation the protein will have 6 histidine residue in its C terminal end. That is the idea. Okay, so that means before insertion of the cDNA to the vector, we will going to add short stretch of nucleotide with our gene of interest. Okay, in such a way that after translation inside the host, it will give rise to a protein where 6 histidine residue will be in the C terminal position. Okay, we all know that protein have to not a main, more specifically I should say peptide have a N terminal residue, another one is C terminal residue. So in the C terminal residue, that is the part where COOH group is located. So in that part we should have 6 histidine residue. Why you need we are going to discuss. So then we will simply use ligase to attach our gene of interest along with the vector to ultimately lead to the production of recombinant DNA. Recombinant DNA, remember, recombinant DNA is, that simply means vectors along with our gene of interest. Okay, so then we will simply use heat shock and CaCl2, that is the transformation procedure, and we will have our transformed colonies, right? Sorry, remember one thing, that we have the ability to distinguish between our transform colonies from our non-transform colonies with the help of blue white screening that here we are using. So that is well, I am not going to repeat this again. So here now in the transform colony we have LAC promoter. And now another important thing that I have forgot to mention, that remember here not only the cloning product or clone product is necessary, instead we are going to express our protein right inside the host. So what we need to do, we need to insert a promoter. So here the promoter that we are using is the LAC promoter. Okay, so along with the LAC promoter we must have to include lactose or isopropyl thiogalactosidase, although the second one is mostly preferred in the medium. Okay, so that LAC promoter will work well and it will give us large amount of the encoded protein. So isopropyl thiogalactosidase, we have already discussed that it is a kind of gratuitous inducer, right? So it will induce the action of LAC promoter and give us large amount of the encoded protein. So in that way we will have our encoded protein, that was our in large amount, that was our main goal, right? So what we have done till now, we have identified the protein that we want to produce in large amount, and then we simply derive, or we can say produce, the cDNA molecule from the mRNA molecule which is responsible for coding of the protein. Okay, now once the cDNA is obtained, we simply add a stretch of the short nucleotide. We can easily add them with the help of terminal deoxynucleotidyl transferase along with the nucleotide TNTPs that we want to add. Okay, so we will going to add this in such a way that at the end, when we have our protein, there should be 6 histidine residue at the C terminal. That is the main goal. Although I have not discussed why we are doing this, I am going to discuss. And our protein will be designed in such a way that it should have a LAC promoter, because remember here our goal is to produce or express the gene, right? So this is a kind of expression vector. Second goal is to produce there should be a multiple cloning site, so then we can easily insert our gene of interest along with origin of replication for the replication purpose and ampicillin resistant gene, that is primary selection marker. And within the MCS there should be a omega peptide, okay, omega peptide of LAC gene. So why we have incorporated omega, not only omega peptide, that means more specifically the omega peptide coding gene should be there, okay. So why we have inserted omega peptide coding gene in the position of MCS, if you want to know, just go back and find out the insertion and inactivation video, you will easily find it out. So now we will add the restriction endonuclease to treat the MCS, so that we can easily insert our gene of interest here, and then the insertion will be performed with the help of ligase, and we will have our recombinant DNA, and this recombinant DNA will be transformed, as here we are using E. coli as a host cell, so we can simply prefer heat shock along with calcium chloride, okay. So after that transformation procedure, the LAC origin of replication will facilitate replication, along with, due to the presence of LAC promoter, transcription will going on, and after that translation will occur, and which will give us the production of protein molecule. Now the fact is, when you lyse the cell to insert the protein molecules, there will be the mixture of endogenous E. coli protein molecules, or I should say the endogenous prokaryotic protein molecule, along with your eukaryotic protein molecule, okay, that is your protein of interest that you want to isolate. So how can you purify your eukaryotic protein or your encoded protein from those endogenous E. coli protein? So the idea is very simple. That is the reason why I have incorporated short nucleotides in such a way that will give ultimately in the protein product 6 cysteine residue at the C terminal, because here we are going to treat our protein, that means total protein sample, with a affinity matrix that is consist of chelated nickel atoms, okay, these chelated nickel atoms. So these chelated nickel atoms have affinity towards this 6 histidine, okay, but another kind of another remaining E. coli protein molecule do not usually get bind with that chelated nickel atoms present in the affinity matrix. So the remaining protein will simply eluted, and then we will, now the question is how we can isolate the bound protein from that affinity matrix. So idea is very simple. Then we will simply decrease the environment of the pH. Sorry, I have made a mistake. So we will decrease the pH of the environment, okay. So once we decrease the pH of the environment, the protein molecules that are bound with the affinity matrix containing chelated nickel will come out, and we will have our protein, a purified protein product, which is usually in large amounts. So that is how E. coli, LAC and T7 promoter based vector are used to produce protein which is usually in low abundance. That is the overall concept behind the E. coli, LAC and T7 promoter based vector. Remember, two main goals. One, we should have a appropriate promoter regarding the host we are use, depending on the host we are going to use for the expression, along with we should have a appropriate cDNA molecule, so that will give us the protein product upon expression. And another important thing that you need to know, that you should have a appropriate purification step, okay, so that you can easily purify your product. Now let's discuss this theoretically. Okay friends, so the first step here is in producing large amount of low abundance protein. That is our actual goal, to produce low abundance protein in large amount, is to obtain a cDNA. The first goal of this should be is to obtain a cDNA. Let me change the color. Okay, is to obtain a cDNA clone. encoding the full length protein by standard methods. Okay. Now the second step is to engineer plasmid vector. Now the second step is to engineer plasmid vector that will express a large amount of the encoded protein when it is inserted into the E. coli cell. That means here we are going to choose our vector on the basis of the host cell that we are going to use, right? And the plasmid should have the promoter that is suitable in that case. So here the vector is obviously the expression vector. So here we are discussing this whole funda with the help of GCSF expression that I have already discussed with you diagrammatically. So that is the idea. So in this case, E. coli transformed with plasmid vector containing GCSF encoding cDNA and LAC promoter. That means the vector that we are going to use should have a LAC promoter along with the other important things that it should have to be a appropriate vector. Now expresses GCSF transcription from this LAC promoter, that is the reason. And if you want to produce your protein at a high rate, then you should incorporate isoropyl thiogalactosidase or lactose in the medium, so it will give you your desired product at a large amount. Now even large quantities of desired protein can be produced if we will complicate our expression system. Now our goal is to purify our eukaryotic proteins. So the common technique that I have discussed with you, that you will modify your cDNA in such a way that the expressive protein will have 6 histidine residue. So here we are going to use short nucleotide to the end of the cDNA. Now the protein modified in this way bind tightly to affinity matrix that contain chelated nickel atom. So I have to discuss with you that we will simply treat our entire protein sample in a affinity matrix containing chelated nickel atom, where no E. coli protein will bind to such matrix, right? So the bound protein can be released by simply decreasing the pH of the surrounding medium, and in most of the cases the procedure yield fully functional protein, pure recombinant protein, right? Because remember, adding a short amino acid residue in the N terminus, C terminus does not affect the biochemical activity of the actual protein. Okay, so we can simply adopt such kind of technique to purify our protein samples. So that is the overall idea regarding E. coli LAC T7 promoter based vector. Question will come in such a way that they will ask you that there is a low abundant protein that is usually in very small amount, and you have to produce this in a very large amount, such as in industrial level. So design a procedure that you can use to produce this kind of low abundant protein in large amount. So you have to discuss this with the help of that particular protein. Main important thing you should have you have to keep in mind that here you are wanting to express your gene of interest, so you have to go for expression vector, right? So here a promoter should be used, that is very important, otherwise the entire answer of you will be wrong. That is the important thing. Another thing is as simple as a conventional cloning technique. So we have discussed our fifth topic of this week also. Now we have only two left. One is expression vector that I am going to discuss with you within this week. Another one is shuttle vector. So till then, bye bye.